I'm Jean Gard. I'm from the U.S. Department of Agriculture in Athens, Georgia, United States. The opinions I'm about to express are mine and do not reflect formal policy by the U.S. Department of Agriculture. My laboratory does a lot of genetic analysis, and I happened to stumble upon a region of the genome that was able to reliably tell us the serotype of Salmonella. And because it was such a small region, we can sequence it by just a standard method. And that allowed us to build a database on Salmonella serotypes that I can then transfer to scientists whoever wants it, for them to do their own serotyping. We've also transferred it to industry so that someone in industry can go out and examine their own farms without having to inform regulatory agencies or to expend a lot of money that typical classical serotyping costs. So it's a way for a farmer or an industry to constantly examine their own ecology when it comes to Salmonella. The method involves a standard PCR reaction, and you generate a sequence that's between 250 and 530 base pairs. And the sequence is specific to the serotype, and then through a database that we have associated with the classical databases, we've built up information so that all you have to have is that short sequence and you can tell your serotype. And what that does is it greatly reduces the cost and also speeds up the rate at which you can get serotype. We basically see the cost coming down about tenfold. This enables us to go and examine larger numbers of samples from farms, and then we can communicate that information very easily through computer and standard software. You do not even have to have advanced DNA analysis tools. So it doesn't necessarily take the place of every other serotyping method out there. It's more of a rapid screening method that can be high throughput, so you can get your information, and if you need to get a whole genome sequence of a source strain, you can, and it's going to tell you which strains to sequence. It's cost savings, it's rapid. It will allow scientists all around the globe to communicate with each other. We're working on about our 10th country interested in the method, and some industry in the United States is already using it, and some pharmaceutical companies. Now, in regards to how it relates to food fraud, someone who's committing food fraud doesn't care about food safety, and a Salmonella outbreak, as food is mishandled and mixed and adulterated, will be very likely to happen. And perhaps a food safety outbreak may be the outcome of someone abusing a food product through food fraud. We're hoping that the method, which is called intergenic sequence ribotyping, or ISR for short, the data is so discrete and so small, we're hoping to make it a cell phone app that a producer or even a farmer on a larger farm could then get weekly updates on what his Salmonella flora is on the farm. There are about 20 different Salmonella serotypes that are of more concern than the others. If you have animals, there will be some low-level existence of Salmonella out there, and what the farmer needs to know is when the more dangerous pathogens show up, or when there's a sudden change in his flora on farm, it could signal a change in the animals that should be followed up. It will also really help the farmer determine if vaccination of his animals are in order. Vaccination is expensive. It may have to be tailor-made to the situation, and this will help pinpoint what kind of vaccine to make and when to give it. And even the marketers, large grocery chains, could ask for a Salmonella report from different farmers. And it's this kind of technique that can be done with so much less expense and so much less nebulous interpretation of the data that will clear up communications between a farmer, a marketer, and even public health facilities that are in the community measuring Salmonella in people.